This is a lecture on cell metabolism and how cells get energy for Bio 101 from September 21st, 2020. And in today's set of lectures, we're going to talk about metabolism. We're also going to talk a little bit about enzymes, what they are and how they work. We're going to talk about the energy currency of the cell, as well as how your cells make energy using oxygen through cellular respiration, as well as how they can produce energy without oxygen. So basically, energy is the ability to do work. And our cells constantly need a supply of energy in order to basically do their metabolic reactions, or the metabolism. And what metabolism is just all of the chemical reactions that take place inside of a cell. And metabolism includes two types of reactions, some that use energy and some that release energy. And there's a balance between reactions that use energy and those that release energy. And sometimes those that release energy, that energy is actually used to do other reactions. And all of these different chemical reactions make up your metabolism. Typically, a metabolic pathway or a reaction contains many steps. And some of these steps involve the making of molecules or synthesis, and these are known as anabolic pathways or anabolic reactions. Anything that's anabolic makes a molecule. And so something like this image seen here, where there are small molecules, individual green circles, and they're built into a larger molecule here. Anabolic reactions require the input of energy in order to take these individual subunits and make them into something else. And there are also some types of reactions known as catabolic reactions or catabolic pathways, where large molecules are broken down into smaller ones. So reactions that break down molecules are known as catabolic. And in the process of breaking down a big molecule into smaller subunits, energy is released. And so anabolic pathways make molecules and require energy. Catabolic pathways break down molecules and release energy. And since anabolic reactions use energy and catabolic reactions release energy, they're actually usually coupled together. And the energy that gets released from a catabolic reaction, where one big molecule is broken down into smaller pieces, is then recycled or reused to power an anabolic reaction, where energy is required to take small molecules and build them into a larger one. And so this coupling of the release of energy from catabolic reactions to power anabolic reactions is one way that your cells can sort of recycle that energy and use it to survive and do all of the necessary reactions for their metabolism. Most of the time, the metabolic reactions, whether catabolic or anabolic, would not proceed very quickly. And that's unfortunate, because if we had to wait for these reactions to happen, we would probably die waiting. And so there needs to be a little bit of help added into metabolic reactions, whether they are catabolic or anabolic, to speed them up, make them happen faster, just so that we don't die while we're waiting for these reactions to take place. And there's a particular type of protein called an enzyme that's function is to speed up or to catalyze these reactions, to make them happen faster. And what's nice about enzymes is that they're not consumed in the process of doing that. You can recycle an enzyme many, many times. It can be used to speed up or catalyze the same reaction over and over again. And this is advantageous because enzymes as proteins would take a lot of basically resources and time to make. And so if we had to make new enzymes every time we wanted to do a metabolic reaction or catalyze it or speed it up, our cells, that would take a lot of time, and our cells would probably die waiting for those enzymes to appear. And so it's nice that you can take an enzyme and basically recycle it over and over and have it perform the same function many times. But what is a little bit unfortunate about enzymes and the fact that they are proteins is that their activity can be affected by the conditions within your cells. And since enzymes are proteins, their function or their activity is determined by their structure. They need to retain a specific shape in order to work. And temperature and pH can affect that shape. Typically very high temperatures, as well as extreme pHs, can what's called denature those enzymes or adjust their shape. And when their shape is messed up, those enzymes are no longer as active. And so there's an optimal or a perfect temperature, and there's an optimal pH or a perfect pH for every enzyme. And fluctuations in that temperature or pH can alter its activity. It doesn't necessarily have to be an all or nothing relationship. Sometimes enzymes just work better at a certain temperature or pH, and sometimes they won't work at all except in very specific conditions. And that is determined by what type of enzyme it is. And enzymes are specific in the type of reactions that they can catalyze. They can only work on specific molecules. And there's a relationship between an enzyme, which you can see in gray down here, and the molecule that that enzyme acts on, or its substrate. So there's a very tight relationship between enzymes and substrates. And enzymes can only work on their specific substrates to catalyze those reactions. And the area on the enzyme where the substrate binds to is known as the active site. And that's what confers the specificity between enzymes and their substrates. Active sites usually have a specific shape that only their specific substrate can fit in and bind to. And when that happens, you can see the substrate here in green binding to the active site on this gray enzyme. They create an enzyme-substrate complex, and then that enzyme can catalyze the reaction and turn this green substrate into blue and yellow products. And when we really get into the most scientific way of discussing what enzymes do to speed up chemical reactions, we're really going to talk a little bit about what's called activation energy. And so the activation energy for any reaction or any metabolic pathway is the energy that you need to start the process, or the energy input you need to start a reaction. And you can see here the activation energy in purple of a particular reaction. And you notice that you have to input a lot of energy before products are generated. What enzymes do is they actually lower the activation energy. And so this red dotted line shows the same reaction with the addition of an enzyme. And you can see that that hill, or that amount of activation energy that's needed for reactants to become products or for the reaction to proceed, is much lower. And that's because enzymes usually bring the substrate and maybe potentially two substrates together in their active site. And by doing that, they increase the chances of a reaction happening. Because you can imagine that molecules are all floating around inside the cell. And if you want to put two of them together, the chances of them just sort of finding each other by chance is low. And that might be why this activation energy in purple is so high, because those two molecules just can't find each other. When you add an enzyme into the mix, it gives both of those molecules a place to bind to the active sites of the enzyme, and therefore increases their chance of finding each other and a reaction happening, and lowers the activation energy as a result. And so since enzymes are so efficient and so good at speeding up or catalyzing reactions, I want you to take a minute and pause the video and just take a minute or two to think about how you can stop an enzyme. Pause the video, take a minute, think about that, and we'll talk about it a little bit. So several ways that you might have thought of are to change the temperature or the pH, which is something that we discussed earlier in the lecture. Both temperature and pH affect enzyme activity, and you can either increase the temperature to really high and denature an enzyme and stop it. You can change the pH from the enzyme's optimal pH and stop it from working. And additionally, you can add what's called an inhibitor to the process. And so you can see here an enzyme reaction between this Pac-Man shaped enzyme and its green substrate. And that green substrate fits really easily into the active site of this enzyme, binds, and then ultimately makes this square-shaped product. And this square-shaped product can actually come back to that same gray enzyme and bind in another spot, changing the shape of the active site, and now that triangle-shaped substrate doesn't fit anymore into the active site. And this actually inhibits or stops that enzyme from being able to work as well. This is what's known as feedback inhibition, because the product of a reaction feeds back on itself and then stops that reaction from proceeding further. You can also have a kind of different inhibition where there's another triangle-shaped molecule that's not the substrate that binds here into Pac-Man's mouth and stops the substrate from binding. That's known as competitive inhibition because you have substrate and another triangle-shaped molecule competing for the same active site on an enzyme. And that's another way you could stop it. But ultimately, what I want you to take from this is all of these different things, temperature changes, pH changes, these two different types of inhibition, whether it's competitive here or kind of feedback inhibition with this yellow square, all affect the shape of the active site. Whether you're changing it because of the end product binding, whether you're changing it because of the high temperature or pH change, whether you're blocking the active site with a competitive inhibitor, you're basically stopping that substrate from being able to bind the active site, and if you do that, you can effectively stop an enzyme. And so I want you to take a second and just go through this question and choose which of these statements is false regarding metabolism, metabolic reactions, and enzymes. Okay, and so the correct answer is C. This is the false statement. Catabolic reactions break down molecules rather than make them. But it is true that enzymes can speed up reactions by lowering their activation energy. It's true that anabolic reactions, the ones that make molecules, require an energy input. And it's true that substrates bind to their enzymes at the active site. And so the only one that's false is C.